All right, good morning, everybody. Feel free to turn on your videos or enter any questions into the chat. I will be asking for some participation and interaction with you via video to just unmute yourself and talk about some of the things that I'm sure you're familiar with. And I'll also ask you to participate in some poll everywhere questions. So that link is pollev.com/SCCR1. It'll be on a slide as soon as I share my slides. I think I'm familiar with most of you on the call, but I'm Kiera Davis. I am the Associate Director for Education and Training in the Stanford Center for Clinical Research. I've been at Stanford now for 6.5 years. Previous to Stanford, I worked as a pediatric cardiac ICU nurse, both at Duke and then traveling around the country as a travel nurse for a few years. I also have a bachelor's degree in history. So combining medicine and history today is probably two of my favorite things to talk about. So I'm really excited about this class. This is the first time we've done this particular class for a GCP topic and presenting on this topic today. So I'm really excited to be able to do this. I want this to be really interactive. So again, if you have questions, raise your hand, unmute yourself, put it in the chat. I don't think we're going to spend an hour and a half on the subject matter, so we'll have some time to talk a little bit about this. So I'll go ahead and share my screen so you guys can see my slides. Okay, so again, the title today is the history of human subjects research, and really some of the agenda or our objectives for today are understanding how we got where we are when it comes to principles, guidelines, regulations, really all of the GCP and the regulations that we follow today for human subjects research came out of something, right? There was some sort of historical example of what not to do in clinical research with human subjects, and it's led us to what we currently use. I will provide you with some additional resources or additional reading at the end of the presentation. But if you have never read the Nuremberg Code, or the Belmont Report, or the Declaration of Helsinki, or the Beecher article. A lot of these things I'm going to talk about today, and we're going to go over them, but I really encourage you to read them. They're not long documents. It really should only take you a half hour to read all of those documents. So those will be resources for you, but if you're working in clinical research, you should know these documents. You should at least be familiar with them. And again, I would encourage you to at least look at them. To know what they say. And a lot of these documents that I'm going to talk about today, they have guided us. They guide us in our clinical research endeavors. They guide us to do the right thing. Many of them are ethical principles in dealing with human subjects. So again, we'll go over them in greater detail in the presentation, but I just wanted to make you aware of those documents and obviously strongly encourage you to read them because they are really important and they're short, so it shouldn't take you a lot of time. So today we're going to talk about what exactly is human subjects research, and obviously that's the acronym for that is HSR. We'll talk about exemptions to human subjects research. We'll go over some examples. We'll talk specifically about clinical trials. We'll talk a lot about the historical background, and if you've attended the informed consent class, then you're probably going to see a few slides that are similar or the same, because I talk a lot about the history of human subjects research in the consent class as well. But I have a few other newer examples in terms of a presentation in today's talk. And then I tied in the 13 principles of GCP to this talk, because again, a lot of our ethical principles, good clinical practice, that is really tied to a lot of these documents. So I bring in and go over those 13 principles so we can kind of see that better connection. And then there's a few questions at the end just to kind of test your knowledge and see what you learned in today's presentation. So again, here's the link, and I know Susan put it in the chat, so it's great if everybody can participate, and it makes the hour and a half go by a little bit faster if we're all interactive here. Okay, so if we are doing research in the United States, there are a few different agencies or entities that oversee clinical research. So you guys are familiar with the Food and Drug Administration. Their code of federal regulations is 21 CFR 50, which is the protection of human subjects. If you are working on drugs, the kind of committee or center within the FDA that focuses on drugs is the Center for Drug Evaluation and Research. Similarly, if you're working on biologics, it's the Center for Biologics Evaluation and Research. And then finally, if you're working on devices and radiological health, that's CDRH, the Center for Devices and Radiological Health. So again, all of these groups, and actually there's multiple groups within each of CDER, CBER, and CDRH. So if you're working on vaccines, you're going to be working with a particular group within CBER. If you're working on drugs, obviously, and different types of drugs, there's different groups within CDER as well. So CDER obviously ensures drugs are safe and effective. Biologics, CBER is regulating biological products for human use. And then CDRH is continued ensuring that devices and radiological health are safe, effective, and the devices are high quality. The other agency is the Department of Health and Human Services. That Code of Federal Regulations is 45 CFR 46. This was originally produced in 1981 with a revision in 2018. The office within the Department of Health and Human Services is the Office for Human Research Protections, OHRP. And this is, they kind of do similar things when it comes to auditing or ensuring that research done at Stanford University is of quality and human subjects are safe. So they're ensuring quality and safety very similarly to what the FDA would do. So if we're doing any government research that's supported by the Department of Health and Human Services, OHRP can come to Stanford University. Same thing, obviously, with the FDA. OHRP is part of the Office of the Assistant Secretary for Health and the Office of the Secretary of Health and Human Services. And again, they provide leadership in the protection of rights, welfare, and the well-being of human subjects involved in any research that is supported by the Department of Health and Human Services. They also indirectly oversee human subject research through local IRBs. So what exactly is human subjects research? And we have a couple of definitions here. The human subjects research, the human subjects definition is per federal regulations, and I'm just going to read this. So a human subject is a living individual about whom an investigator conducting research obtains either data through intervention or interaction with individuals or through identifiable health information. The definition of research provided by 45 CFR 46 is a systematic investigation, including research development, testing, and evaluation designed to develop or contribute to generalizable knowledge. And per the Belmont report, which we'll talk a little bit more about in later slides, the definition per the Belmont report states that the term research designates an activity designed to test the hypothesis and permit conclusions to be drawn. Research is usually described in a formal protocol that sets forth an objective and a set of procedures to reach that objective. The NIH also has its definition. So the National Institutes of Health defines human subjects research as research involving a living individual about whom data or biospecimens are obtained, used, studied, analyzed through interaction, intervention, or identifiable private information is used, studied, analyzed, or generated. It's a wide range of types of research. So we could have behavioral research, other interventional research, pilot or feasibility studies with some of those studies have to do with medical devices, development of drugs and exploratory of drugs, And then mechanistic. And mechanistic is their studies or tests designed to analyze the biological or chemical events responsible for or associated with an effect that is observed. So here's our first poll everywhere question. Of the list below, choose one that is not an example of human subjects research. So if we're interviewing participants, conducting a survey, collecting data on taste and food quality, conducting a focus group, or collecting biospecimens from a participant. I'll give you guys just 10 or 15 seconds. Okay, so I don't know how many people participated. I can't see the actual number, but most of you got this right. So collecting data on taste and food quality is not considered human subjects research. Every other item obviously on this list is considered, is an example of human subjects research. All right, second question, true or false? Secondary research using identifiable information or biospecimens is considered human subjects research. Okay, we're going to talk a little bit more about this in 2 slides. This is actually false. So again, I don't know how many people are answering, but everybody who participated obviously answered true. So this is actually an exemption to human subjects research. We're going to talk about this in a couple slides as well. So here are a few examples of human subjects research. So obviously, if we're collecting biospecimens or blood, that is considered human subjects research. Again, conducting a survey, if we're changing a participant's environment and in observing that change or that reaction, administering any sort of medicine or investigational product, interviewing participants, administering a psychological test, collecting data, conducting a focus group, or testing a new educational technique. So again, this is per NIH. These are all examples of human subjects research. And next, we're going to talk about some of those exemptions. But first, I want to bring your attention to the first good clinical practice principle. So as it relates to this slide. So there are 13 good clinical practice principles. We're going to go over all of them in this presentation, not in detail, but to make you aware of what those principles are. So these are just linked. There's a list of them. They're not in any particular order in terms of how it's presented when you go to the ICH GCP website. But I have touched on all of them in the presentation today. So the first principle is clinical trial data. So all clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting, interpretation, and verification. So I understand, obviously, this is a very broad guideline. It's a very broad principle. But we have, each institution that conducts research is responsible for doing these and setting up these processes. If you're following federal regulations, you're following GCP principles, you're in good shape. But this is one of the first principles that I wanted to mention linked to this example of HSR. Okay, so let's talk for a few minutes about some of these exemptions to human subject research. There are 8 categories. The first is if research is conducted in an educational setting using normal educational practices. So if you're doing a new educational intervention, that would be considered human subjects research. But if you are observing behaviors or any other collection of information from normal educational practices, that would be considered an exemption. The second is using educational tests, surveys, interviews, or observations of public behavior. So limited IRB review may be required for this, but again, this is an exemption to human subject research. The third one, benign behavioral interventions in adults. Again, limited IRB review may be required, but obviously benign behavioral interventions don't pose any sort of risk to human subjects. So again, an exemption. Okay, so this isn't quite the example. I think that's 8 actually, but 4 has to do with secondary research as well. So secondary research, so that's research that is not obviously the primary reason for doing the research, but secondary to the main hypothesis of your research question. And it's information that has been kind of collected and collated, and you're using that set of data for a secondary purpose. So this is so even if it's identifiable information or biospecimens, if publicly available or recorded, such as subjects cannot be re-identified. So we recently had a question about a study where the PI was looking to collect biospecimens for the purpose of a biobank related to COVID. And we had to clarify with them, well, what kind of biospecimens are you collecting, and is that information identifiable? So if you're going to just biobank these specimens and use them for future research, we need to make sure, one, that's listed in the consent form correctly, right? And we need to make sure that if this is going to be used by others in the future, it has to be in the consent form correctly, and we need to ensure whether or not this information is identifiable, because that will obviously affect the consent form. The fifth one, public service program research or demonstration projects, and we'll go over some examples of these on the next slide. Taste and food quality evaluations, back to that first poll Ev question. Storage or maintenance of identifiable information or biospecimens for secondary research use. So this would require broad consent, and limited IRB review would be required. And that goes back to that same example that I just provided. And then the second POLEV question was about this. So secondary research using identifiable information or biospecimens. Again, broad consent, limited IRB would be required. IRB review would be required. So some examples of these. So when we talk about educational setting using normal educational practices, so these are examples of these are effectiveness of online training as supplement to regular instructional approach, testing the effect of biweekly science-focused field trips on middle school student learning. So again, all exemptions. Educational test surveys, interviews, or observations of public behavior. So you want to conduct a focus group of adult community members to discuss access to dental care. Or you want adults to complete a questionnaire about outdoor exercise, including collection of participants' age and zip code. If you're collecting any PHI, there will be some limited IRB review, but again, an exemption to human subject research. Benign behavioral interventions. So you're studying young adults, evaluating their preferred snack foods following a television program. A study among adults investigating text versus voice message appointment reminders on annual physical appointment attendance. I should have put the 8 categories on my slide so I don't have to go back and forth. Secondary research using identifiable information. So this would be an example, would be collecting random samples of patient de-identified data every 6 months for medical records, or a collaborator removes an aliquot of blood from coded samples. Aliquots are relabeled to a random non-linked code. So that would be obviously your de-identified data. Public service program research or demonstration projects. So this example of this is the study of barriers to obtaining new Medicare benefits or outcomes assessment from government-sponsored mental health services. Taste and food assessments. So you're evaluating wholesome food preferences or looking at approved levels of an agricultural chemical and the taste of vegetables. Storage or maintenance of identifiable information, sorry. So the researcher has created a data set containing identifiers from a previous study to conduct future research. Again, like I mentioned, this would need to be in the consent, and you would need to go through a limited IRB review, or if you're saving blood samples from a collaborative study for a future research question. So that was my example related to the biobank for biospecimens related to COVID. And then secondary data, the last example, using a data set from prior study containing identifiers to answer subsequent research questions, or using blood samples from a collaborator study for an additional research question. Okay, so next question, just a yes or no question. A study involves the analysis of de-identified blood samples of patients with disease X who were treated with an approved drug to evaluate the level of a protein in the blood associated with the therapeutic effects of the drug. Is this a clinical trial? One more seconds. Okay, so this is not a clinical trial. So these patients were treated with an approved drug. So we're not looking at any sort of investigational new drug. All of the samples are de-identified, so they cannot be linked back to a participant. And the research is just looking to see if that drug, affects this protein in the blood. So again, de-identified information, it's an approved drug. This would not be considered a clinical trial. What about this one? Yes or no? A study involves recruitment of participants to receive a chronic disease management program to determine the maximum required sessions. Is this a clinical trial? Yes. And what does anybody want to tell me why this is a clinical trial? What tipped you off? Everybody got it right.
Recruitment of participants.
Yeah, right. So you're recruiting participants. There's no information. There's nothing in this question about de-identified information. You're assigning them prospectively to this disease management program to determine its effectiveness or how many sessions are needed. Yeah, so this is a clinical trial. And so we're gonna, there is this decision tree that Susan put together for this presentation. This is from the Office of Human Research Protections to kind of help us determine what is a clinical trial and what is not. This is really for you as a resource. I'm not going to go through all these yes or no questions, but it's a decision tree to help us determine if something is research. Sorry, this isn't for a clinical trial. This is a determination of research. Here's another decision tree. This is for the National Institutes of Health to help determine if something is a clinical trial. So this one's pretty short, so I'll go through it. So the first question is, does the study involve human participants? So obviously no, not a clinical trial. Yes, we move on to the next question. Are participants prospectively assigned to an intervention? If it's yes, we continue on. Is the study designed to evaluate the effect of the intervention on the participants? Moving on to the next question, is the effect being evaluated a health-related biomedical or behavioral outcome? So if all of those are yes, this study is considered a clinical trial per NIH definition. Okay, any questions or comments so far before we move into some of these regulations and ethical principles?
Hi, Kiera. I have a quick question.
Yeah.
So as far as like health data, so let's say that you had, I'll give you an example. Actually, on the Janssen study, there was a sub-study within the vaccine study that the sponsor wanted to look at medical records for 5 years prior to the vaccine and then 5 years after. And to see how like their health kind of was influenced and long-term outcomes. So technically under the sub-study, I guess there wasn't an intervention because they're just looking at health data, but it was de-identified. But there was like an intervention, which would have been the vaccine. So would that be considered, could that be considered human subjects research?
What was the IRB determination?
I don't remember.
Okay. Yeah, I think I would need a little bit more information to be able to answer that. But because if there's an intervention and you're following a participant for 5 years, then that would more than likely be a clinical trial. Even if the information is de-identified. And that doesn't really make sense to me if the information is de-identified. Maybe prior to, but prospectively, following them for 5 years, they wanted to de-identify the information. Interesting. Okay. That sounds like a clinical trial. I don't know why they would want to de-identify the information, but I'm happy to talk more with you about the details of that, just so we can both better understand it. But if it's a prospective assignment of a vaccine and you're following the participant, then that sounds like a clinical trial to me. Okay, so let's talk a little bit about these regulations and ethical principles. So this slide is taken from the consent presentation. We talk a lot about these in the consent class, but really the first code of principles that anybody in the world saw was the Nuremberg Code. So this was created in 1947 following the trials at Nuremberg after World War II. The Nuremberg Code was created by 3 people, I think 2 Americans and 1 German, all in the field of ethics and in the field of medicine. And upon hearing about the atrocities committed, by these Nazi physicians, they put together this list of 10 principles to follow when conducting research with humans. And we're going to go over that list, but I wanted to give you a little bit of historical context. The next document, again, these are principles to follow, is the Declaration of Helsinki. This was created by the World Medical Association in 1964. Most of actually all of our GCP principles are based on the Declaration of Helsinki. So again, another landmark, really important document. And again, we're going to talk more specifically about it. I'm just giving you a little bit of an overview. The Belmont Report was written in 1978, really following the Tuskegee syphilis study. And there were various steps for why the Belmont Report came into being. And again, we'll talk about that in a couple slides. I've talked already about the FDA CFR and the Department of Health and Human Services CFR. We've talked a little bit about ICH GCP, which was created in the early 90s and was revised most recently in 2016. And then HIPAA, which was created in 1996, which is obviously for the confidentiality of PHI. That is also obviously a regulation that we follow related to protection of PHI and confidentiality of PHI. So here's the timeline that I'm going to more or less go over with you today. Again, this is adapted from the consent lecture with a few other new historical examples that I'm going to go over, including the Willowbrook State School Studies, the Jewish Chronic Disease Hospital Studies, San Antonio, Texas Contraception Clinic Studies, and I'll go over a couple more of these, like the Tuskegee syphilis study. We talked a little bit about the experiments during World War II. So again, really the first half of the century, you see all these really horrible examples of what not to do with human subject research. And then in the second half of the century, we see these ethical guidelines created, regulations, GCP, and really, again, what led to what we follow today when it comes to ethical principles and regulations. So diving in a little bit more from the previous slide, we're going to talk more about these examples in the coming slides. So I'll talk briefly about the Nazi experiments on prisoners during World War II, the first congressional federal law regarding protection of human subjects, these three examples that kind of turn the tide to better guide us in ethical principles and regulations were the thalidomide tragedy, the creation of the Declaration of Helsinki, and the Beecher article. And then we'll talk about Tuskegee, Willowbrook, Jewish Chronic Disease Hospital, and the San Antonio, Texas Contraception Study. And this is just another timeline kind of showing much of what I just discussed. Okay, so I'm sure you guys are at least a little bit familiar with the medical experiments that took place during World War II, but wanted to go over those with you again. So these were experiments dealing with Jewish prisoners, the Roma people, as well as any person who was imprisoned during World War II by the Nazi regime. They were studying various things. They looked at experiments dealing with the survival of military personnel. So they wanted to know how to save the Axis power soldiers, right? So they conducted experiments with high altitude to determine maximum altitude from which crews of damaged aircraft could parachute to safety. They conducted freezing experiments on prisoners to find an effective treatment for hypothermia, and they tested various methods of making seawater drinkable. They also conducted experiments to test drugs and treatments. This included immunization compounds and antibodies for the prevention and treatment of contagious diseases, such as, let me get to my notes page here. Such as malaria, typhus, tuberculosis, typhoid fever, yellow fever, infectious hepatitis. Prisoners were exposed to mustard gas, bone grafting, and were tested with new sulfa drugs. The picture on the left there are a picture of one of the Mengele twins. So Dr. Mengele was particularly interested in comparing some of these experiments on twins. And he also did multiple sterilization experiments. And then the final example was really any experiment that was to advance Nazi racial and ideological goals. So after World War II, only a few of the biomedical experts who helped to implement and to legitimize the Nazi racial hygiene policies were ever indicted or disciplined professionally. Many, unfortunately, continued their careers. Of course, some of them were charged and were actually put to death after the Nuremberg trials, but many who were also involved, there was really no punishment for those individuals who participated. So again, the Nuremberg Code was written in 1947. These were 10 ethical standards for allowable medical experiments post-World War II. And again, this was really the first document that had ever been created for human subjects research. This is one of the cornerstone and foundation documents for other ethical principles and regulations. And we're gonna go over them now. So the first one is voluntary informed consent of human subjects is essential. Experiments should yield fruitful results for the good of society. The experiment should be based on animal studies and knowledge of the natural history of disease. The researchers should avoid all unnecessary physical and mental suffering and injury. No experiment where there is an a priori reason to believe death or disabling injury will occur. There must be a favorable risk-benefit analysis to avoid unnecessary pain and suffering. Researchers must properly prepare, proper preparations must be made to protect participants from injury, disability, or death. The experiment conducted must be done by scientifically qualified personnel. Participants have the right to withdraw without restrictions, and the scientists must be prepared to terminate the study at any time, obviously, if there are any safety issues related to the experiment. So again, at the time, there were really no laws, regulations, codes, or formal documents that stated which ethical principles researchers needed to follow. This code articulated the basic requirements for conducting research in a manner that respects the fundamental rights of participants, and it helps to ensure their safety. The US, like I said, I would think it was two US, I think they were both physicians. Maybe one of them was a physician and one of them was an ethicist, but I think there were two Americans that had an integral role in the creation of the Nuremberg Code. And this, I think in a way, eventually led to some of the federal regulations. But at the time, again, the United States had no federal regulations related to human subjects research. We had a couple of acts related to the Food and Drug Administration. The Food, Drug, and Cosmetic Act came later. But there were some congressional acts related to drugs, specifically drugs, but it really, again, didn't help to protect the right, safety, and welfare of human subjects. So the first congressional law protecting human subjects actually arrived in 1955. This was at the university, the kind of event that took place was at the University of Chicago. Researchers decided to audio tape jury deliberations of a criminal trial in Wichita, Kansas. They wanted to study the jury's decision-making process and whether showmanship by the trial lawyers influenced the decision of the jury. The jurors were not told they were being recorded. The researchers didn't want to influence their behavior, so they kept that secret. And after there was a study published and discussions were focused, so the study was published, the information was published, and a lot of people said, hey, this doesn't really seem right. So there were discussions on the unethical, nature of this study. It was deceitful. These were supposed to be closed conversations, private conversations among the jurors. Privacy and confidentiality were supposed to be protected and expected by the jury. And so after this example, Congress passed the first federal law prohibiting the recording of any jury deliberations. So this was the first congressional kind of example to protect human subjects. So in the late 1950s, the use of ethics review of research began. And this was after the creation of the National Institutes of Health and what was called at the time the Clinical Research Center. So the Clinical Research Center's policy required review of all research by an ethics committee before study initiation. Okay, I mentioned this a little bit earlier. I talk about, I think actually all of these in the consent class as well, but these three events really kind of turned the tide and led us down the path where regulations, the regulations that we follow today. So the first is the thalidomide tragedy. And again, if you attend the consent class, we do talk about this briefly. And I think I'll do the same thing here today. If you, just to get a little bit more participation from you guys, does anybody want to raise their hand and tell just a brief synopsis about what the thalidomide tragedy was and what happened?
Yeah, I did a paper in grad school on the pharmaceutical industry, and thalidomide was one of the examples I used. So this drug, I could be off on some of these facts. It was a long time ago, but I think it was in the 60s. This drug was approved for pregnant women to use to help sleep, and it resulted in the children being born with very deformed limbs. I believe there was some issues with the pharmaceutical company and the data, but I think what you're trying to get at is pregnancy and issues with human subjects. That's my summary.
Yeah. Thanks, Joel, for participating. And Diana, you commented, was that the medicine for pregnant women that caused birth defects? Yes, this was a teratogenic medication. As Joel said, it was used actually to treat, morning sickness, so nausea and vomiting in pregnant women. And this drug actually did not make it to market in the United States. There was her last name was Kelsey. She prevented the drug from coming to the United States because she wasn't comfortable with the data. She's like, we're getting this to pregnant women. I don't think this is, there's not enough data to support its safety. So we're not going to approve it here in the United States. So that one decision, protected a lot of women, a lot of children. But in other parts of the world, primarily, I think this was in Europe, these women were given this drug, right? They were told this was going to help you with your morning sickness during pregnancy. And at the time, obviously, they did not know that it caused teratogenicity. So it caused fetal demises. It caused preterm birth, a lot of congenital heart defects, genitourinary defects, and most notably and probably most common were these upper limb deformities. So at the time, it was not standard practice, nor actually a requirement to inform patients of the investigational nature of a pharmaceutical being tested. So this came later with our regulations, but it was used in the 50s. And like I said, this affected probably thousands of children, as well as fetuses who did not survive. So this actually took place over a couple of years, and by 1962, it became very apparent that thalidomide was what was causing these deformities and children to be born with complications, and it led to this public outrage and outcry of, wait a second, shouldn't someone be protecting these children, these women, and human subjects in general? And this one example changed the Food and Drug and Cosmetic Act to include consent for investigational products. So at that time, there was no requirement to include that information in a consent form. But due to obviously the results from thalidomide, that became a regulation through the Food, Drug, and Cosmetic Act. So the second event, and we're going to talk about this on the next slide, is the Declaration of Helsinki, and then I'll talk more about the Beecher article as well. Okay, so the Declaration of Helsinki, again, was created in 1964 by the World Medical Association. The WMA met in Helsinki, Finland. This is why it's called the Declaration of Helsinki. And they drafted this document outlining what they thought should be the standards for human subjects research. And again, this was one of basically the second landmark document, which led to many of our regulations and other principles, such as the Belmont Report. And these are the main bullet points from the Declaration of Helsinki. Again, this is not a very long document, so I encourage you to just read and be familiar with what's in this document. But essentially, much of what is in the document is obviously presented on this slide. So again, these were ethical principles for medical research involving human subjects. The Nuremberg Code was used to build on or was used as a document to help form the ethical principles of the Declaration of Helsinki. One of the general principles is stated, it is the duty of the physician to promote and safeguard the health, well-being, and rights of patients, including those who are involved in medical research. Looking at the risks, burdens, and benefits, researchers must provide careful assessment of predictable risks and burdens weighed against foreseeable benefits. And really, if the risks and burdens outweigh the benefits, this is not an ethical study and should not move forward. Vulnerable groups and individuals should receive specifically considered protection. Scientific requirements, and there should be scientific requirements as well as a research protocol for research involving human subjects. Research ethics committees review and approval must be provided. The privacy and confidentiality of personal information must be obtained, and voluntary informed consent must be provided. And the ones that I have bolded here are also linked to our GCP principles. So again, we talked about clinical trials, collection of biospecimens. The second one, second principle outlined, second GCP principle outlined in the Declaration of Helsinki is ethics. So clinical trials should be conducted in accordance with the ethical principles that have their origin in the Declaration of Helsinki and that are consistent with good clinical practice and the applicable regulatory requirements. The second one is trial participants. So the right, safety, and well-being of the trial subjects are the most important considerations and should prevail over interests of society, of science and society. Trials should be of good quality, so good quality trials. Clinical trials should be scientifically sound and described in a clear, detailed protocol. Compliance with the study protocol. So a trial should be conducted in compliance with the protocol that has received prior IRB or IEC, Independent Ethics Committee approval, and obviously should have a favorable opinion. Sixth one is medical decisions. So the medical care given to and medical decisions made on behalf of subjects should always be the responsibility of a qualified physician or, when appropriate, a qualified dentist. And then the seventh one is confidentiality. The confidentiality of records that could identify subjects should be protected, respecting the privacy and confidentiality rules in accordance with the applicable regulatory requirements. Actually, that was the last one. So that third event that kind of, again, helped kind of progress and move us forward from a regulatory standpoint is the Beecher article. So Dr. Henry Beecher was an anesthesiologist at Harvard. He studied the placebo effect and was really interested in how research was being conducted in the United States as it relates to human subjects. He collected detailed information on about 22 studies where he found, really egregious examples of unethical research practices. He brought that to the New England Journal of Medicine to publish, and the editor of the New England Journal of Medicine worked with him to kind of reduce that number. It was, the article would have been, pages and pages and pages, and it's kind of a lot of information. So the editor worked with him to bring that information, basically providing information on 12 studies, maybe the most egregious of those 22 examples. And so this really brought to light through publication the unethical practices, including the lack of informed consent with human subjects, and multiple examples of increasing the risk to participants. This was really a pivotal moment in history of human subjects because this article was published by a member of the research community who was conducting research and focused on the need to improve the ethical conduct of human subject research. The article pointed out the unethical research practices on human subjects and led to many ethical principles, including peer-reviewed. So research must be peer-reviewed. This led to the development of the Code of Federal Regulations for in the United States. IRBs must be used. And then it eventually led to the National Commission for the Protection of Human Subjects. So Beecher helped to, again, usher in this broad transformation of bioethics and the patient-doctor relationship to shape medicine's code, which helped Congress establish the National Research Act of 1974, which developed the National Commission for the Protection of Human Subjects. And we're going to talk about the Belmont Report in a couple slides. Okay, so how are we doing on time? We're doing all right. The 4 studies that that I would like to hear from you guys, if you're familiar with, one, I know someone has to know about the Tuskegee syphilis study, but these other 3 studies were actually, I knew a little bit about them just through getting my master's in clinical research. So 2 of them I had learned about in my program, but the last one with the contraception clinic studies was new to me. But if you know about these examples, it would be awesome to hear from some of you. And I see a few familiar names. I'm gonna, if nobody volunteers, I'm gonna call on you.
Hey, Kiera.
Hey, Chris.
Hey, is the Willowbrook State School Study, is that the twins?
No. Any other help with that one? We'll talk more about it, obviously.
Yeah. This was Jewish twins. I can't remember, and I know this is completely offline, but they were separating them, and then they would do testing at each one of their families. They were adopted out to different families to see.
Oh, that's another example, Chris. I don't know
right.
about that one.
It was, nature versus nurture, and families that were fluent and others that were poor. So that one was really interesting, and I'm happy to talk about Tuskegee.
Yeah, why don't you go ahead and give us a little synopsis of Tuskegee?
So Tuskegee syphilis study blows my mind that when it went on as long as it did. This was very poor Black families, Black men in the South, Mississippi, Louisiana, and they did not treat them for syphilis, even though we had penicillin, and they wanted to see what happened to them and to their partners. And it was just horrific that it went on for so long when there was a cure.
Yeah, exactly. They studied, they actually didn't even tell them they were studying syphilis. So it's like the Department of Public Safety or something within the US government wanted to understand the natural progression of syphilis, right? So what happens with these people, with these men particularly who have syphilis. So in the 1930s, there wasn't a cure, but penicillin was developed in the 1850s, and it was found obviously to be the cure for syphilis. So again, as Chris said, it should have been given to all of those participants in this study, but they withheld that as treatment for 20 years for some of them. And so the natural progression of syphilis is quite terrible. You actually, tertiary syphilis, you develop neurological symptoms and eventually will die from those neurological symptoms, encephalopathy, and just really horrible death. So yeah, this went on for 40 years. President Bill Clinton finally paid reparations to the families in the 1990s to say, wow, we really screwed up. The US government's very sorry. Please accept this, this payment as our apology. But it affected, a lot of people actually in Alabama. It was in the South. They were all sharecroppers from Alabama. And they not only were not consented, they weren't told they were part of a research study. They were told they were being treated for bad bloods. They were being provided with kind of free medical care in a sense, but obviously that medical care was not good. They did not provide that penicillin to them. So yeah, thanks, Chris.
Mm-hmm.
Okay, so we'll talk. So the Willowbrook State School studies, which is this picture here. So this took place in the 1950s. Investigators at this school intentionally infected mentally challenged or developmental delayed children at an extended care facility in New York with hepatitis. So they were investigating how transmissible is hepatitis. And they actually fed them a solution made with the feces of those with active hepatitis. Obviously, there was no sort of consent for this. Obviously, taking advantage of a very vulnerable population who cannot consent to this. I don't even think the families were made aware of what was going on in the state school here. In the 1960s, the Jewish Chronic Disease Hospital, which is pictured here, this is when live cancer cells were injected into the bloodstream of chronically ill. Most patients who had cancer, mostly were demented, and they were injecting them with live cancer cells. This took place in a New York City hospital. All subjects had some form of illness that compromised their immune system, and the study was designed to determine the influence of a weakened immune system on the spread of cancer. And then finally, I have one more picture here. And finally, in San Antonio, Texas, this took place in the 1970s. So obviously, each decade we're seeing just this horrible, egregious examples of human subjects research. Even after following, we have by this point, some of them by the 1960s, we have the Declaration of Helsinki in place. Obviously, the Nuremberg Code is in place, and researchers are continuing to not, follow those ethical principles. The contraception clinic studies in the 1970s took place in San Antonio, Texas, and these were studies. This was a study that was looking at contraception clinics serving mostly indigent patients, and they wanted to evaluate the efficacy of some of these contraceptive pills. So they evaluated the efficacy of various types of oral contraception in a randomized, blinded, placebo-controlled study. So right from that statement, a placebo-controlled study, what does that tell you about what some of these patients are receiving, right? So they're going to a contraception clinic specifically to obtain contraception to prevent pregnancy. And these researchers put some of these patients on placebos. So they thought they were taking oral contraception. And obviously, none of the participants were informed about the study and that they were participating in a study. And it led to, many unplanned pregnancies, right? Because of women receiving placebos. Okay, so a little bit more about the National Research Act of 1974 and the development and the use of IRBs. So the National Research Act led to the creation of our modern IRB system. The federal regulation 21 CFR 56 required that IRBs approve human subject research. And this, the National Research Act defined the policies and procedures that IRBs must follow when considering what is research, and obviously benefit and risk, safety and well-being of participants, and obviously collection of quality data. They help to establish the criteria for the IRB approval of research proposals. So after these four examples that we just went over with Tuskegee, Willowbrook, the contraception studies, the Jewish Chronic Disease Hospital, people became aware of what was going on as it relates to human subjects. And these four examples really led to additional public outrage and increased the national debate about human subject research. The consensus was reached that federal oversight of human subject research was required to protect the rights and welfare of research subjects in both biomedical and social science research. And it led to this National Research Act of 1974, which led to the National Commission for the Protection of Human Subjects in Biomedical and Behavioral Research. That committee developed the Belmont report, and we're going to talk about that in the next slide. The National Commission was composed of experts in ethics, religion, law, industry, and medicine. They defined problems, issued recommendations, and published numerous reports before the kind of final Belmont report was published in the 1970s. And in 1978, that final document was the Belmont report. So again, these are fundamental principles and guidelines for human subject research, setting boundaries between practice and research. And who knows what the three, before I click on, who knows what the three ethical principles are that are in the Belmont report? Or just name one of the three.
I can never say it. But what is it?
Beneficence. Yeah.
Thank you.
Yeah, that's one. Beneficence. Does anybody remember the other two?
There's one that's about, it begins with a J.
Yep, justice.
Thank you. That was a tough one to remember how to pronounce.
And the third one is respect for persons.
There you go.
So, so respect for persons obviously has to do with individual autonomy. This is an implementation of respect for persons is one way is with informed consent. So respect for persons acknowledges the freedom and dignity of subjects and requires subjects to voluntarily consent to participate. So again, I talk about this a lot in the consent course, obviously. We have additional protections for vulnerable populations. If someone is not able to consent, obviously we have legally authorized representatives, or we have assent for those who can sign assent. But this has to do with letting people make their own decisions, right? So this is the prevention of coercing someone into participation, whether you're the clinician or whether you're a family member. This has to do with individual autonomy and respect for someone's decision to participate or obviously not to participate in research. Beneficence is maximizing benefits and minimizing harms associated with research. So this helps to ensure that risks are reasonably justified. And as I stated earlier, if the benefits or the harms obviously outweigh the risks, the harms outweigh the benefits, this would not be an ethical study. If we know that it's going to be harmful, if we know that this intervention will cause, horrible things or even death, obviously that is unethical. They should, researchers should not proceed, with that study. The third, excuse me, the third principle in the Belmont report is justice. So justice is the equal distribution of risk and benefits across society. So this means that you need to be equitable. There has to be equitable selection, recruitment, and fair treatment of research subjects. And then a little bit more about justice. Members of society who are likely to benefit from the research should also bear the potential risks of the research equally. Research should not select specific classes of subjects because they are readily available. So can you guys think of an example where participants were readily available to study? They were, conducting a research study on someone because of who they are is also another. We actually just went over the example, but I hope I'm making sense. I'm trying to get you guys to kind of apply this information to past examples. Yeah, Randy, raise your hand.
Yeah, so for the example of, say, like the Willowbrook study, right? I mean,
Sure.
those kids were right there.
Right there, vulnerable population, absolutely.
But I'd also like to elevate that, it kind of goes the other direction nowadays, where when we think about doing trials equitably, and and this is the work that I'm doing, that I'm interested in doing, we are we really actively seeking out those populations that we're applying these interventions to, or are we going to who is easiest to reach?
Yeah.
For example, when we look at interventions for diabetes, and most of the folks that are coming into the trials are white, and who's most impacted by diabetes? That's just one example.
Yeah, no, that's a great example. It's a great example. And all of these examples, right, with Tuskegee, for instance, or Henrietta Lacks, that significantly negatively impacted the Black community from a research perspective, right? So the Black community does not want to participate in research because of what the government has done or what other researchers in the past have done, specifically with the Black community. The Tuskegee syphilis study is really the example that I'm talking about. But I think that's a really good point, and that's why it's so important to diversify our recruitment as much as possible. I talked about this in a recent presentation that I did. So if you go onto the FDA website, there is information on every drug related to sex, race, and ethnicity, and how those drugs may affect race, ethnicity, or sex differently. But when you look at the breakdown of race and ethnicity in these studies, overwhelmingly, most of the research participants are white. Very small percentages of Asian, very small percentages of Black, very small percentages of Hispanic population. So it's Randy, it's exactly right. So it's mostly white people, and this is why it's so important that we focus recruitment efforts on non-white people. Yeah, really good discussion there. Thank you, Randy, for that. Okay, so let's move on to GCP principle number 8. So this has to do with trial risk and trial benefit. So this is very, in sync, obviously, with our second principle here of beneficence. So before a trial is initiated, foreseeable risks and inconveniences should be weighed against the anticipated benefit for the individual trial subject and society. A trial should be initiated and continued only if the anticipated benefits justify the risks. Just a couple more bullets here, specifically about respect for persons. So participants must voluntarily consent to participate in research. The consent must be informed consent. Also really important why everybody takes the informed consent class that Susan or I teach. Participants' privacy and confidentiality must be protected, and participants have the right to withdraw from research participation without penalty or repercussions. This has to do with our GCP principle number 9 of informed consent. So freely given informed consent should be obtained from every subject prior to clinical trial participation. Here are our 8 essential elements of informed consent. Again, this is part of the consent class, but many of these are part of the ethical principles that we've already talked about, with benefits and risks and discomforts. You must acknowledging that what they're participating in is research. You must list alternative treatments, that you'll maintain confidentiality of records, compensation and treatment for injury if that were to happen, contact information that their participation is voluntary and they can withdraw at any time. Okay, so for the remaining GCP principles, I couldn't specifically link them to some of the examples that I provided previously, so they are just listed here. But 10 through 13, obviously, with the first one on this page, is information on the medicinal product. So the available non-clinical and clinical information on an investigational product should be adequate to support the proposed clinical trial. And that's why it's so important, obviously, that we understand the development of say it's a drug, understand people are following good manufacturing practices, people are following good lab practices, that the information that's coming from a lab prior to human subjects research is trustworthy, and that any animal research supports the use in humans. And there's good, you know, X practices, right? So for GXP, for anything that is involved in any investigational product or research, there are good practices for that, including good lab practices. GLP. Trial staff. So each individual involved in conducting a trial should be qualified by education, training, experience to perform his or her respective tasks. So this has to do with all of us. So are we qualified to provide informed consent? Are we qualified to I'm trying to think. I think it kind of starts with understanding the history of human subject research, right? So do we understand where we are today and why we are here today with the regulations that we have? So, and then of course with those who have licenses, clinicians, RNs, MDs, RTs, anybody who's participating in research, they have to be qualified by license as well. I talked a little bit about GMP, so good manufacturing practices. This is another principle of GCP. So any investigational product should be manufactured, handled, and stored in accordance with applicable good manufacturing practice. They should be used in accordance with the approved protocol. And then finally, the last GCP principle is quality assurance. So systems should exist with procedures that assure the quality of every aspect of the trial and should be implemented. So I have a couple slides that I included for IRB requirements. I'm going to skip over that just in the interest of time. I was going to go over this if we had time, but they will be provided to you. So I just wanted to include really what the IRB requires as it relates to human subject research, and then going over what the different submissions are for IRB, full board review, expedited, or exempt. And then I also listed some of the most common forms of noncompliance and violations. So despite all of the ethical principles and even regulations that we have in place, these are the most common forms of noncompliance. So one of the first ones is deficiency in presenting the essential elements in the ICF to our research subjects. Failure to document in the ICF, inappropriate enrollment procedures with possible undue influence, inadequate written procedures or communications to conduct clinical research, including SOPs or formal trainings, research that is initiated without IRB approval, insufficient information provided to the IRB, protocol changes without IRB review. So the researcher just goes on and decides to implement changes to the protocol without IRB review. Suspension or termination of IRB approval and the study continues, failure to conduct continuing reviews with the IRB, changes to the risk profile, that information is either not shared with the IRB or is not shared with research participants. Same thing with significant new findings. Proper monitoring is not conducted, reporting of adverse events is not done, and of course, just general non-adherence to the protocol. Okay, so for the remaining time, we'll have some kind of test your knowledge questions, and we'll wrap up and we'll have some time for questions or any other conversation about this topic. So which three events led to the congressional hearings and federal oversight of human subject research that we covered today? Yes, you guys paid attention. So C is the correct answer. All of these are important. I don't mean to take away from their importance with this question, but the three specific events that I went over are the ones that kind of led to the public becoming more aware of research practices or inappropriate research practices or unethical research practices, which led to Congress becoming involved. Okay, which event helped to enact our current IRB system of research oversight? Willowbrook State School Studies, the National Research Act of 1974, the Nuremberg Code, or the Beecher article? Now that I think about this question, I think there's actually two right answers. B and D. And actually the one that I was going after is actually D. So the Beecher article helped to kind of usher in the use of IRBs, but the National Research Act of 1974 was also pivotal. So B or D for this question. I'll change that for the next class. This should be more explicit. Which landmark document offers 3 fundamental principles for the ethical conduct of research: respect for persons, beneficence, and justice? Yes, this is a softball question, the Belmont report, which we went over pretty in depth. True or false? Human subjects research will always require full board IRB review. So we didn't have time to go over the IRB review, but good question to know the answer to. So this is false. Human subject research will not always require full board review. It will depend on the study. It depends on the study. There's some information in those slides, but the answer is false. Which CFR regulates IRBs? So is it part 50, part 54, part 56, or 45 CFR part 46? All over the place on this one. The correct answer is actually C. Nobody got it. 56 is for IRBs. So 21 CFR Part 50 is human subjects research with the FDA. 45 CFR Part 46 is human subjects research with Department of Health and Human Services. 21 CFR Part 54 has to do with financial conflicts of interest. So 56 is the CFR that regulates IRBs. Okay, this is my last slide. It has some additional resources and information on there. As Susan stated earlier, she will send out these slides to everybody. We have about 10 minutes. I'm happy to discuss anything that we talked about today or answer any questions. Thanks for attending and for your participation. This was a really fun topic for me. So I hope you guys learned something, feel a little bit more prepared with your understanding of the historical background of what mostly the United States has gone through in terms of regulations, but certainly these examples, some of these examples obviously happened outside of the US. So like Susan said, please complete the evaluation that she'll send out. And if you don't have any questions, obviously you guys can head out. Thanks, Randy. Thanks for your participation. Thanks, Chris. Thanks, Joel. Thanks, Diana. Thanks, Christine.
Hi, Kiera, it's Olivia. I have a question. I'd like to come back to the secondary, use of samples. So you said that is not considered as human research, but however, sometimes they do require IRB approval. Could you comment on that?
Yeah. So for instance, it will require IRB review, and you have to list in the consent just like a very broad a broad explanation of what those samples will be used for. And so the participants have to consent to that. And IRB also must approve that. So it's not like it's just a free for all, but it's not considered to be human subjects research. It's an exemption to human subjects research. So.
Right. No, I understand that part, that you need to make the patient aware of the potential use of their samples for future research. But I'm trying to understand is when you use those samples, and remarkably, when you even say they are not de-identified for future research, you need to create a new protocol with the IRB to get approval.
Yeah, so I mean, again, it doesn't mean that it doesn't mean that it's IRB exempt. It's just an exemption to the definition of human subjects research.
Okay, I thought that identifiable information was human research, was and not de-identified. So
it's secondary. It's secondary research, either using identifiable biospecimens or identifiable information, or not. Right? If it's de-identified, that's an easy sell. It's not considered human subjects research. But if it is a data set where the information is still identifiable according to the definition provided by the NIH, it's not an example of human subject research. It's an exemption to human subject research. And if it's an exemption to human subject research, the IRB does not require as much information, but the IRB may still want information on what you're doing with those samples. You can't just take the samples and do whatever you want with them. And there's been some dialogue and conversation about that. The example I provided is with the biobank that one study or one researcher was trying to use. So there are specific requirements that you have to list in that protocol or in the consent form. You can't just say, even if it's secondary research, you can't just say, are you willing to provide your biospecimen for the purposes of A and B, and maybe we might do something else with it in the future. We don't know. That won't really be allowed. So the IRB has to agree to what the secondary research samples will be used for, and obviously the participant has to agree to what those samples will be used for as well, and whether or not they'll be identifiable. Is that any more clear?
Yeah. Well, thank you. Yeah. Thank you.
Okay. Yeah. No problem. It's a little confusing. I agree. Any other questions? All right. Well, we will end the presentation here. Again, thanks for your attention and participation. And if you have improvements or comments about the content or how it was presented, we're all ears. This is really the first time we've done this, so we're happy to take some feedback and make the next course that much more beneficial. All right, thanks everybody.
Thank you. Thanks.